and have those who are starting to continuing to join us do so as we get things going. So thank you again for joining the Scintica instrumentation event. My name is Tanya Coulthard, and I will be your host for today's webinar. Our webinar today is entitled A Cytosmart Product Review, the Lux2, Lux3 FL, and the Omni Live Cell Imaging Systems. Today's webinar will be given by Dr. Katie Parkins. Dr. Parkins is an application scientist here at Scintica Instrumentation, focusing a lot of her time on the various products that we offer, mostly within our imaging group of products. Katie does hold a PhD from the University of Western Ontario in medical biophysics and completed a postdoctoral research fellowship at Johns Hopkins University. Throughout her research training, Katie has focused on the development and application of novel molecular imaging tools to monitor and treat diseases, specifically focusing on cancer. Katie has used imaging extensively in her research and has been a tremendous resource to her lab mates and now our customers about how to truly get as much out of our imaging systems as possible. Today, Katie will share with you as much of her experience as she talks about the Cytosmart products. She will turn her attention to live cell imaging, what it is, the advantages of being able to image cells in real time, and how they are interacting with their surrounding environments. She will then move to describe the Lux and the Omni products, and finally discuss some common applications and share some of the related imaging examples. Before I turn things over to Katie, just a few housekeeping items. We do anticipate today's webinar to be about 40 minutes in length, leaving us plenty of time at the end to address some of the questions that you may have. Please do use the Q&A dialog box at the bottom of your screen to ask any questions throughout the presentation, and I'll work with Katie to address as many of those as we can towards the end of the time we have together. If we are unable to answer all of your questions, we will work together to pull together a transcript of those questions which are unanswered, and we'll send that out to everyone over the next week or so. Please also note that we are recording today's session, so if for some reason you lose connection or you can't hear everything clearly, or you simply wish to share that recording with your colleagues, you will have access to the video file later. And now, without further ado, I'd like to turn things over to Katie for today's webinar.
Thanks, Tanya, and welcome to everyone joining us for today's webinar. As Tanya mentioned, today's session is focused on the Cytosmart product line that we offer here at Scintica. Many of you joining us today probably have heard of either one or all of the live imaging systems I'm going to be discussing, but the goal today really is to give a clear overview of the similarities and differences between each system so that for those that are interested, we can start to think about which option may be the most relevant for your particular applications. In terms of outlining today's topics, I want to start with a brief introduction to live cell imaging, what it is and why you would want to add it to your lab's capabilities. We'll talk about the advantages of live cell imaging compared to some more conventional methods. I'll then go into each system individually, starting with the Lux2, the Lux3 FL, and finally the Omni system, highlighting the system specifications, showing some example images, and then finally discussing some potential applications for each system. Okay, so jumping right in. So what is live cell imaging? Live cell imaging is the study of living cells using time-lapse microscopy, and it's used by scientists to obtain a better understanding of biological function through the study of cellular dynamics. Now, both CO2 and tri-gas incubators are designed to generate and maintain a certain environment, and they do a great job of imitating physiological conditions so that cells can grow in their ideal environment. The problem really lies in more so the frequency of the door of these chambers being opened when cells are removed from the environment every time a measurement is made or an image is taken. And this is usually done with an external microscope. What this means is that most scientists never actually measure cells under physiological conditions, and the cells they're looking at are generally in some state of shock. As a result, in recent years, live cell imaging has become really a desired and often necessary tool in many laboratories to more accurately monitor and measure cell dynamics under the appropriate environmental conditions. So what are the advantages of live cell imaging compared to using something like a traditional microscope? Well, of course, it's going to be less time consuming. So your microscope can sit within the incubator and run your experiments remotely, which frees up a lot of time for you to do other things in the lab. It enables longitudinal studies. So this is an important one. We can start to study dynamic processes rather than just collecting snapshot images at particular time points. We can acquire images of cells while they're under their preferred conditions, so there's no risk of them going into a state of shock. You can monitor your cells remotely. So this has become an extremely valuable feature, especially during COVID, where groups may want to limit the amount of people within the lab at one time or limit the amount of experiments taking place. And finally, these systems are designed to be easy to use and quick to integrate into an existing lab compared to traditional microscopy that typically requires either an expert or a trained personnel to acquire the desired images or videos. So that leads me to the Cytosmart products. So the entire product line that I'll be discussing today has been designed with kind of the same goal in mind, and that's really to develop a highly compact, easy to use, and affordable system for bright field and fluorescence live cell imaging. Each of the three products has two components, the microscope itself, and then the software, which is all done on the Cytosmart cloud. Many of you are familiar with the names of the microscopes, but we'll start with the Lux3. So this is the smallest of the three options. Then we have the Lux3 FL, just slightly bigger than the Lux2 and offering fluorescence imaging capabilities. And then finally, the Omni system, which is large enough to scan an entire cell culture vessel, but still compact enough to fit on the shelf of most standard incubators. Okay, so the Lux2 is really kind of the original mini microscope offered by Cytosmart, with the initial goal of allowing researchers to monitor their cells throughout their entire culture period without taking the cells out of the optimal culture environment. In terms of specifications, I think it's important to really give you a sense of the size we're talking about here. So it weighs just over a pound and fits quite easily in someone's hand. So I said small, and I truly meant it. Groups having lab space that extends over a number of different areas or research buildings can really appreciate this. So you can very easily just unplug the system, carry it or put it in your backpack, take it across campus with you, and basically set up shop somewhere else in a matter of seconds. So very different than a traditional microscopy system that's set up in a core facility. In terms of magnification, it's equivalent to a 10x objective with up to 20x digital zoom. Something to note is that the field of view is fixed. So this particular microscope is just able to take images or time-lapse movies of one region of your flask. So you can move it yourself to another area, of course, but you're not able to stitch those images together since you're manually moving the flask, and it would be nearly impossible to get it to the exact position needed. That being said, you can take as many images as desired for as long as you want within that set field of view. In terms of workflow, you can see here in the video, the idea is that the Lux system would stay within your incubator. You simply place your flask on the microscope when you want to start an experiment, and then everything else is done from the computer. So this would allow you to either image or monitor your cells over time from wherever you like. Currently, you can choose between two different image analysis algorithms. So the first is confluency. So here we can see, we can look at cell division over time, or the scratch or wound healing algorithm. So in this case, the software can automatically determine where the scratch is and determine the healing of the wound or the migration of the cells over time. If you acquire a time-lapse movie, images are automatically analyzed using either one or both of these algorithms. However, you can also choose to download the images. So some prefer to use their own method to analyze their images on something like Fiji, Cell Profiler, or MATLAB, for example. In terms of potential applications, the most common is going to be things like imaging cell division, looking at stem cell differentiation, growth dynamics, looking at cell migration or wound healing, or studying colony formation or chemotaxis. So here I'm showing some example images of HepG2 cells for up to 47.5 hours after seeding. And you can see quite nicely the size and the number of colonies formed by these cells can be monitored over time. The Lux has also been used for monitoring cells in microfluidic chips. So you can see in this video, the setup is quite ideal. There's lots of space to position the slide on top in the middle, and then the tubing will drape over the sides of the system with the liquid going in. Here's an example of what this type of data can look like. So this movie shows neutrophils migrating through a microscopic maze. So neutrophils are the type of white blood cells that migrate towards the place of infection, basically to clean up the infection. So this maze was developed by Dr. Ramia of Harvard to investigate the effect of sepsis on the speed and efficiency of the neutrophil migration. So the neutrophils enter the maze on the left side, and then they migrate towards a chemoattractant in the reservoir on the right side. So using the Lux2, they can analyze how efficient, basically whether it's a straight line or a random line, and how fast the neutrophils move towards the attractant. Now, just recently, Cytosmart has launched the Multilux Auction. This is to take advantage of just how small these systems are. So you can now have up to 4 Lux2 microscopes connected to a single computer. So this really provides opportunity for long-term comparison studies where you have multiple conditions on side-by-side systems, or just an ideal setup for a larger lab group, since a single experiment can happen over a period of days. So the system may be unavailable to a particular user. You can see here in the software, when you go to set up your experiment, you can choose your device in the top left corner. So you see device 1 through 4. And then you can set the relevant parameters for each independently. Okay, so now we'll move on to the Lux3 FL. So this system is just slightly larger in size compared to the Lux2. It has the same bright field capabilities, but it also has additional red and green fluorescence imaging capabilities. In terms of specifications, you'll see it's very similar to the Lux2 with the same magnification, the same operating conditions, very similar field of view, and then same culture vessels that can be used. The main difference, as I mentioned, is going to be the red and green fluorescence. So we can see here the excitation and the emission filters that are used. And these have been chosen basically to allow for the most standard red and green probes to be used. So for example, TD tomato, GFP or RFP, tracker orange and tracker green. For those that are interested, I do have a list of compatible dyes and fluorophores, so I'm happy to share those with you after today's webinar if you are interested. Okay, so why add fluorescence? So to explain this, I like to show this nice bright field video that can be acquired on the Lux2. We can nicely visualize the cells here. There's a bit of division, a bit of movement, but overall not a lot happening over quite a long period of time, about 1 day. If we then watch the same video incorporating fluorescence, we can actually get quite a lot more information out of the same data. So this group in particular was studying CHO K1 cells immediately after some sort of treatment. And cells that display green fluorescence are undergoing apoptosis, whereas red fluorescence in the nucleus indicates that the apoptotic pathway is completed. And images were taken every 10 minutes for the same amount of time. Now, using the analysis software, you can actually get a measure of this over time to show the percentage of red cells versus green cells, or in this case, the amount of cells that are undergoing apoptosis versus the amount of cells that are dead. But you can, of course, design your experiment so that these fluorophores turn on or off depending on the cellular process that you are interested in studying. So what are people using this for? Compared to the Lux2, I think many are using it for viability studies. So either something similar to what I just showed, or using a dye to show when cell death is occurring. Transfection is a popular application. So looking at the percentage of a population that's expressing your gene of interest a certain amount of time following transfection or transduction. And that can be important, especially if you're looking at viral transduction in perhaps a BSL-3 lab where you don't want to remove the viral counterparts until a certain amount of time. You can actually have the microscope keeping track of all this and getting your transfection efficiency data. Co-culture experiments. So maybe you want to label one cell population in red and a second in green and look at how these cells are interacting with each other. So this can be done maybe with cancer cells or immune cells or various populations of neurocells. A number of different cellular processes and nanoparticle work. So many people use nanoparticles that already have a fluorescent tag attached to them. So this is a great application for those interested in looking at maybe labeling efficiency or nanoparticle characterization. Definitely options here. Okay, and then we will move on to the Omni system. So this is a system that's getting quite a lot of attention right now, as it's still quite compact and easy to use, but it can collect a lot of data in a short period of time and is really well suited for groups needing to do full well analysis. So you can see here on the image on the right, it's still taking up, you know, a full shelf of the incubator. So it is quite a bit larger. It's not like the Lux systems where you can kind of put 4 in a row, but it is compact enough that you can fit it within your incubator and leave it there for the remainder of your studies. Okay, so to kind of go through the Omni system, I like to show this video because it kind of highlights the main differences between the 2 Lux options and the Omni. So again, the main idea is that it can sit within the incubator, within the optimum culture conditions. The objective you can see now moves and it will scan the entire plate. And it takes about 6,500 images in 6 minutes. So it's very quick. And this is regardless of what plate or culture vessel you choose to use. But I think many are going to be using the 96 well plates for this, because as you can see, you can get individual well data over time, but you can also analyze all 96 wells at once. In terms of the algorithm, so it's similar to the Lux systems where you can look at confluency, and this can be done again either in individual wells or individual field of view like the Lux, or throughput experiments. You can basically compare the confluency across multiple wells or multiple conditions. You can, of course, also use the scratch assay. So here the green line is showing what the software has picked up as the scratch. And then over time, the scratch area will decrease as those cells start migrating towards each other. And again, you can kind of imagine how much data you're able to pull from this when you're comparing wells in something like a 96 well plate. So this is an example here. So again, it's the same algorithm that's being used for the Lux system. So this is a scratch assay, but definitely more appealing for groups that want to do rapid throughput for more longer term studies. So here, this group is able to look at a number of different conditions and show that as you increase cytokeratin D, you also see an increase in levostatin concentration. Okay, so just comparing these options kind of at a glance. So they are very similar. They're all going to be very compact systems that you can keep within your incubator and maintain those culture conditions. The important thing to remember, the Lux2 is bright field only. So that's the smallest option that we've talked about. The Lux3 is fairly similar in size to the Lux2. It also has bright field, the same bright field capabilities that the Lux2 has. The main difference is that it has the red and green fluorescence imaging capabilities. The Omni system, the main difference is going to be both the size of the system, but also the field of view, because you're able to now scan through an entire culture vessel to get data from either each well or stitch together a field of view for an entire well plate. In terms of the software analysis, it is super user-friendly, which people really like. You're able to essentially get started immediately. As I mentioned, this is a big difference compared to a standard microscopy system where you typically are going to require an expert or trained personnel to operate the system. And then your trainees or lab members may have to book time with that user or get proper training before they can take images. With these systems, you essentially just need to plug in the system. There's two connections. There's something that connects the power to the microscope itself, and then a separate connection that connects the computer to the microscope, and that's it. So there's not actually any buttons on the microscope itself. You simply just connect it to power, and then everything else is done right through the software. You can use all of the channels separately or combine. So again, it's very straightforward to use. You see the lock Z function in the top right. That's going to either, if you want to set your focus or your intensity on one channel and apply those to all channels, you can hit the lock Z button and it will maintain those parameters for all channels. Or you can unlock that feature and you can change them all individually. Of course, one of the major benefits of these systems is that you can monitor remotely. As I mentioned, this has been really crucial during COVID when many of the labs have been shut down. Maybe we've had limited access to the lab, or we're just trying to keep our numbers down in terms of who's using the incubators and who is going through the lab. Cell monitoring is one of those things where you don't necessarily need to be there. You just need to check and make sure that your cells are not overgrown or your transfection is working as expected. So the ability to monitor these things remotely, I think, is a huge benefit. And the Cytosmart Cloud really allows you to do that. So you can just go online and you can access all of your projects in your experiment folders as they're happening and adjust the parameters as you see fit. And then there's integrated image analysis. So similar to how you acquire the images, the analysis is very straightforward and very logical, and it's all done through the Cytosmart Cloud. So you can, again, adjust your channel settings on the right. You have an option to, once the image has been acquired, you can actually export it however you like. So you can keep the bright field image. You can turn on and off the red and green channel in the case of the Lux3. You can look at different measurements. So for example, if you're using the confluency algorithm or the scratch algorithm, it's going to be collecting all of your data and you can see it in real time on the cloud as your experiment is progressing. You also have the option to view the images as it's happening. So let's say you collect, you wanted to collect a time-lapse video so that it takes one image once per hour for 24 hours. You can actually log onto the cloud from home and take a look at the images that have been acquired partway through your experiment to try and see, is this what I was expecting, or do I need to adjust the intensity levels? etc. So that can be really handy. And then you can actually export your video files and images right here from the cloud. So up here, you see the turtle over here and the cheetah. This is going to be regarding the speed of your video. So you can choose to either trim the video if there's only a certain part you want to use for a scientific presentation or a publication, or you can show the whole thing, but maybe you want to slow it down to a certain part or speed it up. So these are all options, and they're all integrated within the image analysis. And like I said, super user-friendly for beginning trainees. Okay, so in summary, all of these systems have a couple things in common. So firstly is the automated image capture. So definitely allows you to save time. You can allow these products to basically sit within your incubator, capture your fluorescent and or your bright field images for you without actually having to be within the lab. Time-lapse movies. So many of the users are not necessarily getting these systems just to collect an image. They really are adamant on getting a time-lapse movie. And that's because it allows you to get the whole story rather than just seeing kind of tidbits of individual events depending on the time points that you're imaging. Red and green fluorescence in the case of the Lux3. So this allows you to kind of expand the number of variables that you can examine and really expand your studies in terms of what you're looking at and the data you can pull from those time-lapse videos. And for those that are a little, maybe intimidated by fluorescence, maybe you don't have red and green cells in your lab now, I encourage you to still reach out. I'm happy to help. There's many either cell lines that you can easily purchase from suppliers that would work well for these systems, or there's also dyes that it's just a quick assay. You add the dye, whether it's for apoptosis or something else, and then you can just image. So there's many protocols that we have in place, and it's fairly simple to implement if you are interested in adding the fluorescence. Integrated image analysis. So I mentioned this, immediate insight into both the area or the object number in terms of the fluorescent cells, or for bright field, just looking at confluency alone. And you can actually set alerts. So I mentioned you don't have to be in the lab to actually image your cells or monitor them. You can actually set alerts if you wish within the software so that you get a text message or an email, when your flask has reached 80% confluency. Or maybe in the case of transfection or transduction, you want to be alerted once 80% of your cells are expressing red or green. So there's lots of different options, and it really can be designed for each individual's application. Incubator friendly, so of course, they're very small. You can work either within the incubator and then the computer. Most people like to put the computer that's going to be setting up your experiments directly beside the incubator. So there's a computer that will be attached to the system, and this is just to set up your experiments, so set your parameters, and then everything else can be done remotely from a personal computer. And these systems are all very plug and play. So as I mentioned, you can get started instantly. Just a very quick training will get you up and running. And when I say get started instantly, we often find when we ship these systems to customers, by the time we reach out to see, have you received your Lux system, they've already set it up on their own and have started taking pictures. It's that easy. So really, really a great plug and play system. And of course, they're all cloud-based systems. So this allows you to inspect your cells anywhere and anytime with no more unnecessary lab visits. And while these systems are kind of all a little bit different in price, depending on your needs and the algorithms that would be most applicable for your lab, they are definitely a very cost-effective system, especially if you consider a Lux2, for example, it's so compact, you can essentially have up to 4 fit within one shelf of your incubator, taking up very little space. And they can all be operated on one computer. So very different than sort of the traditional microscopy system that sits in a core, and you can only have kind of one user per image session. Okay, with that, I'm going to pass it over to Tanya for a quick audience poll.
Excellent. Thanks, Katie, so much for the great discussion so far. I hope everyone's found it useful. I am going to launch our audience poll. We'd just like to get a little bit more information about you following the webinar. If you'd like us to follow up and talk more about your specific research goals and learn more about the Cytosmart products. If you have any other questions specific to your research and how imaging could help you, we'd be happy again to have Katie reach out to you as well. Happy to send you the link to the webinar, and just interested if you're here to learn more about interesting technologies. While we give everyone a few minutes to respond to those questions, if you do have additional questions that you'd like us to work through with Katie, please again use the Q&A dialog box, and I'd be happy to do so as we move into the Q&A session after the poll. Okay, thanks everyone for taking the time, and I'll turn things back to Katie for now, and I think we're moving on to the Q&A.
Great, thanks, Tanya. Yeah, if anybody has any questions, I would be happy to answer those now. I also want to encourage anyone that may be interested in a demo of the system, please reach out, because this is a system, it's nice to kind of see it either in person or remotely to get a sense of both the size as well as, you know, the software and being able to interact with the software and how that works. So please don't hesitate to reach out with any questions or interest in having a demo of the systems.
Perfect. We have a few questions to get us started. And again, any additional questions, please use that Q&A dialog box at the bottom. Katie, you talked about the ease of using the system and checking in on your cells remotely, but do you require the internet to access and operate the system?
Mm-hmm. So that's a good question, and one that we get quite often. I would say the short answer is yes. You really want to have internet with the system to be able to, you know, get the full capabilities of the Cytosmart cloud, because all of your images are going to be stored there. So you do need that internet connection for both the images and the time-lapse videos to get to the cloud, as well as to be able to do the analysis. That being said, if you really didn't want to use internet, you could store kind of a limited amount of data locally.
Got it. Because there may be situations where specific institutions or companies would not want it connected to the internet. And I think we've encountered that once or twice.
Yes, that's right. So you can, the way that the cloud works, it's done through Microsoft Azure. So it's very, very secure. So there's no issues there, but kind of depending on each institution's preferences or each lab's preferences, we can kind of configure it in a way that is going to be best utilized for each group.
Perfect. Another question is, what kind of data can you expect out of the system with regards to your experiment?
Yeah, so in terms of, for example, you can get the confluency data. So similar to the graph I was showing with the red and the green line, that's something you would see with the Lux3, where you can look at object count or percentage of population. So you can look at how many of your cells are fluorescing red versus green or versus negative. And then in terms of the bright field or the Lux2 or Omni options, you can look at the confluency or the percentage of, I guess, the degree of migration over time. So it looks like a graph with the time along the x-axis, and then either the growth is going up over time or the percent area in the scratch is decreasing over time. Now there are kind of those nice tables and charts that I was showing throughout the presentation. Those are all done in the software automatically for you. So you can either choose to pull those directly and use those in your presentations or your papers, or you can just, there's an option, you can just right click and export the raw data into Excel and then put that data into your own formatted table or chart.
Perfect. Another interesting question, is there an option for fluorescence capabilities that scans the entire field of view like the Omni system?
So this is another one that comes up a lot. So this is because obviously people would love to have the fluorescence capabilities of the Lux3 system, but often need the entire field of view scanned, such as in the case as the Omni. So the Omni is bright field only. This is definitely, a longer term goal of Cytosmart for that exact reason. I think it really would be valuable to those doing fluorescence imaging. But I do think at this time it's more of a longer term goal. And that's because it's kind of a tricky problem to solve in the sense that you would be scanning an entire plate, but you don't want to be, exciting adjacent wells at the same time. So I think the short answer is it's not an option at this time, but I think I'm hopeful in the near future it will be an option.
Excellent. There's no other questions that have come in just yet, but I do encourage everyone to just take a few minutes. If there are questions that you have, please use that Q&A dialog box and we'll get to them. At this point, I will thank everyone for their time in joining us today. And also thank you, Katie, very much for the interesting discussion. And I hope that everyone found the information, again, interesting and relevant to the work that you're doing. If in the days and weeks to come you have further questions about the system or you want some more information, please do feel free to reach out to Katie or Scintica directly. Emails are on the screen there, or give us a call. We'd welcome any opportunity to talk about your specific research goals and anything that you'd like to do moving forward. And again, thank you for your time. At this point, there are no other questions, so I think we'll give everyone back the 20 minutes in their schedule, and we can use that hopefully to finish up our day early as well. So thank you for taking the time again today, and looking forward to seeing you at another future Scintica instrumentation event. Thanks and take care.